Dear learners, welcome back to NPTEL online certification course on novel drug delivery systems. I am Dr. Satish Devanapalli, Assistant Professor at Department of Pharmaceutical Sciences and Technology, Institute of Chemical Technology, Mumbai. In our last session, we have started with the introduction to novel drug delivery systems, and we will learn few aspects. We will see the previous presentation, and then we will go further what we have learned in our previous session. So, in our previous session, we have discussed on the what are the drug delivery systems, and then we have seen the conventional and the drug delivery system differences, and then we have seen the evaluation and the terminology. So, here in last lecture we have discussed till common terminologies, and today we are going to discuss few more terminologies, and then we will see what are the advantages, the limitations, and what are the generations of the drug delivery systems. So, here in last lecture we have seen the importance of drug delivery systems, how which is different from the conventional system. With this picture representation, we have learned the effectiveness of the drug delivery systems. And here also we have seen various novel drug delivery systems, various approaches, and here we have seen the various differences based on these aspects, and we have seen the difference between conventional and the novel delivery systems. Here these are the some technology we have seen, and what are the brand name, and then the year of the invention which we have discussed. Here we learn the various control release systems, and also we have seen the difference between the control release and the sustained drug release. Here we learned with some pictorial representation what are the terminology we use in the drug delivery systems. As we have seen, your drug need to be maintained in this level which is called as therapeutic range. So, to achieve that, we must convert our formulation into zero order and controlled release systems. Yes, and the terminology which is not limited to this, there are the other terminologies which also we can be used, which are few things are missed, that is extended release, right, and such terminology which are commonly used in the drug delivery systems. And here today we are going to discuss another terminology which are used in the drug delivery systems. So, here another terminology which is nothing but nanocarrier systems. So, we have a dedicated week for learning of the nanocarrier systems. There we are going to discuss on the various nanocarrier strategies. In that we will discuss on the polymer based nanocarrier systems, and we will discuss on the liposomal formulations, nothing but lipid based. Another third category is inorganic and metal nanoparticles. So, yes, nanoparticles and nanocarriers, nothing but here we use various materials to deliver the drugs from one place to another place, which are nothing but nanocarriers, or we can say also vehicles which will carry the molecule from the administration site to the disease site. So, here we use polymers, lipids, and even we use hybrid polymer and lipids also, and inorganic with lipids also, that is also known as hybrid category systems. And we can also use liposomes and other lipid formulation like solid lipid nanoparticles, and various types of vesicular systems also we use in the nanocarrier systems. Of course, the term itself suggests that the theoretical range of this size is 1 to 1000 nanometer, which are called as nanocarrier systems. by using lipid, polymer and other type of materials, we can convert our drugs into the nanosystems. And yes, the nanosystem, the with that we will see the high drug loading in the carriers, and also it will take the carriers, it will take the molecules into the right site. With this we can also improve the permeability, penetration also. So, this terminology also we are going to use. The extended words are nothing but nano medicines, which are also we can use it while learning the drug delivery systems. So, these are the some of the examples we can see here, the pictorial representation of the nanocarrier systems, liposomes and the dendrimers, which can be prepared by using the polymers, and here the micelles we can also prepare by using various types of surfactant, as well as even polymer micelles also possible by using various types of the polymers. So, another word is that we are going to learn in another week, the targeted drug delivery systems. Now, remember we are going from conventional to drug delivery system, and in drug delivery we are discussing various aspects like control release and sustained release, and we are going to also discuss on the nanocarrier systems, and then we are discussing the targeted drug delivery. So, from moving from drug delivery to target drug delivery, there is a huge differences are there, because as we discussed in our previous session, so if you give any conventional injections, so the drugs will not reach to the target site, which is nothing but non-specific delivery or non-specific targeting. So, with that we will see lot of problems in the patient, especially we know that the chemotherapy, right. So, all anti-cancer drugs we know that which are toxic in nature, which kills the normal cell also. In such cases, target delivery system has a huge demand for the treatment of the cancer patient. So, by using active targeting or by using passive targeting, so I am using two words here, active and the passive targeting. These words also will terminology will use in the drug delivery system. With these two targeting strategies, we can reach into the cancer cells. So, with the non-specific we will cannot reach into the tumor site, and when you go with the non-specific by simple giving the injections and the as in a tablets or capsules and any other formulation, so the drugs will reach into the normal cells. So, their role is the drug will kill the cells, that is called as cytotoxicity. So, it will kill the cells. So, that is leads to the bone marrow depression, and also we will see the alopecia, nothing but hair loss, and the third one will see major side effect in the cancer patients while during the chemotherapy, which is nothing but anorexia, loss of appetite. It will affect on the human body. So, such cases we need to go with the tumor targeted drug delivery systems. So, with this system we can take the molecule into the specific site. So, target drug delivery system is a system of specifying the drug moiety directly into the target area. Either it may be organ, particular tumor organ, or into the particular cells or subcellular level of specific tissues, even into the organelles of the various cells also we can target by using the target delivery system. With that we can reduce the non-specific toxicity, as we discussed three major side effects of the anti-cancer drugs. With this we can reduce the non-specific toxicity of the conventional drug delivery, which is major limitation in terms of the in case of conventional drug delivery. So, thereby reducing the amount of drug required for the therapeutic efficacy. So, you can reduce your dose, you can go for even twice and thrice also. So, here this example we can see here, with this we will understand how these systems are very effective. So, this is a tumor cell. Once you have given injection, right, so it will not reach to the non-specific target sites. Because of these carriers, when you are functionalizing with any type of the ligands, this will definitely it will circulated through all the organs, but which will not accumulate in other normal cells, because the tumor cells have a such structure, so that the small molecules are which are the nano range, which will enter into the tumor site. So, this mechanism is called as EPR effect, enhanced permeability retention effect. So, when you see the feature of the tumor cells, and if you see the micro environment of the tumor, you will see there is a leakage in the lymphatic system, which is called as lymphatic drainage, and also they have the leaky vasculature, so that from this capillary our drug molecules or nanocarriers will enter and it will retain in the cancer cells. So, that is why it is called as enhanced permeability retention. So, this is called as EPR effect. So, which will specifically happen with the nanocarriers when you do the targeting. So, now here going to see what are the advantages of the drug delivery systems compared with the conventional system, as we are discussing, right. So, here the improved therapeutic efficacy of the formulations. So, here once you convert your conventional formulations of your drugs into the novel drug delivery system, and you will see the change in the efficacy. It will depends on the formulation strategies to convert the water insoluble or low permeable drugs into the water soluble, as well as the extensive permeability of your drug. So, with that you will see the improvement in the bioavailability, and that further leads to the high efficacy when you compare with the conventional systems. And not only that, when you do the targeting, your molecule will reach into the target site. And another aspect is that when you use these polymers, when you use this delivery system, you can also avoid the degradation of your molecule by coating or by encapsulating into that carrier systems. So, with that also you will see the improved efficacy compared with the conventional system. And here this is the depends on the release. So, here the time based control and the prolonged release, modified release comes with the polymers. So, here we use such a polymers which will control the release as well as which will protect from the all type of degradation. So, this already we have discussed. So, this is very very important. There are the drugs which require longer time in the serum or in the plasma. Such drugs we need to identify, and then we need to convert into control systems, so that you can reduce the toxicity of the such drugs. So, this already we have discussed. By using the drug delivery systems, we can also reduce the dose frequency. In nowadays, we know that by taking off the long acting injectables, we can also reduce the dose frequency. For example, in one case, if you need to take the dose 3 times per day, right, especially in case of diabetic patient. For example, they will take the tablets 2 times before meal or after meals, they need to take twice a day. Such cases, if you convert your formulation into the modified release, so such cases we can reduce the dosing frequency. So, with that we can improve the patient compliance, and definitely every drug has a side effect. Some of the drug have the major side effects, some of them have the minor side effects, but all the drugs are toxic in nature by the definition. So, with that we can also reduce the side effects by using the drug delivery systems. So, these all are interlinked, each point is interlinked with the another point. So, with this we can reduce the side effects. So, when you perform control release, when you do the changes in your formulations by using drug delivery system, you can also improve the bioavailability because of your absorption will change, right. In the ADME profile, absorption, distribution, metabolism and excretion, these 4 factors are very, very important for the pharmacokinetic studies. So, here if you modify if you improve the absorption of your drug, definitely your bioavailability will increase, and that will depend on the solubility as well as permeability, and other factors also like the enzymatical degree also you can reduce by using drug delivery systems. With all this we can enhance the bioavailability. As I mentioned, we can also protect various the temperature, pH sensitive and harsh condition degradations by using the drug delivery systems. You can encapsulate your drug into the pH sensitive or temperature sensitive polymer, so that your drug will not relieve at that particular site. We can shift from the stomach pH to the colonic or intestinal pH, so that your drug will not degrade in acidic conditions. So, for that we need to take pH sensitive or intestinal responding polymers. So, with all this we can improve the patient convenience and the compliance. So, these factors are very, very important. So, with these advantages, so we need to look for the developing of the new drug delivery systems. So, these are the few limitations of the drug delivery systems. While developing, we need to consider all these factors also, because the onset of drug action is very, very important for the any type of formulation or delivery systems. So, if there is any delay by using various types of the drug delivery approaches, so that will not show the action on that particular disease. So, delaying in onset of a drug action is one of the major limitation. While developing, we need to look for this parameter. And also, yes, possibility of dose dumping in case of poor formulation strategy. So, dose dumping is nothing but the release of the high amount of the drug release will happen at a short time. So, that is also which may one of the major limitation. So, we need to check every interval while designing or developing of this formulation. It may happens with the less understanding on the formulation. So, we need to focus on the drug properties as well as polymer and their response in the biomedium, either it may be any type of biological mediums. So, dose dumping need to be avoided. This is one of the major limitation which happens with the poor formulation strategy. Greater dependence on GI residence time of the dosage form. So, yes, because we need to depend on the when you are making any novel drug delivery system, especially for the oral delivery, and this will depends on the GI time, and that need to be studied while developing the formulation. Possibility of less accurate dose adjustment in some cases. While developing, we need to calculate the doses for the converting from conventional to the into the novel drug delivery systems. So, these calculations and we need to consider, and after making also the release will also affect your efficacy. So, the possibility of less accurate dose adjustment in some cases. So, there are the cases when you are shifting from the conventional to other carrier systems, other nano carrier systems. So, the dose need to be accurately maintained. And the fifth point is that cost per unit dose is higher when compared with the conventional dose. Yes, because here we are going to use new materials and the new process and your product also new. Definitely the arrangement of everything will increase. So, in terms of manufacturing cost, raw material and the maintenance of such products will be definitely different and the cost will go higher compared with the conventional doses. Not all drugs are suitable for formulating into the extended release dosage form. That it is a correct point, but we based on that in our next class we are going to see what are the properties we need to look while developing the control release and other type of formulation. Even in the nanocarrier systems also we are going to discuss. So, one system will not fit for all drugs. All drugs will not fit into the in one system. So, we need to convert our drugs into required format and then we can give it in a as in a drug delivery dosage form. So, here the generations of drug delivery systems. So, here we can see, so you can also say that it is a evaluation of the drug delivery systems. So, we can see it is classified into 5 generations. Okay. First generation and so here we know that these are the conventional systems, nothing but capsules, tablets, emulsions and suspensions. Already we have discussed here the efficacy is low and safety also low and which are not targeted, right, and it will show the maximum side effects. So, these are the some of the limitations of the first generation. Now, in the second generation, modified action systems, enteric coating and repeat prolonged systems. As we know, enteric coating which will release in the intestinal pH, so which will protect the drug molecules from the acidic conditions. So, your biological molecules are pH sensitive molecules that you need to convert into the modified action systems by using enteric coated polymers, other some polymers we can prolong the action. So, these are the second generation. So, the efficacy will improve, right. And now, the third generation which comes in the controlled drug delivery systems, osmotically swelling and diffusion control systems. So, these systems are the third generation systems. So, here also we use various polymers which will help for the drug delivery. And another one is the fourth generation. So, targeted delivery system, yes, we can use target our formulation to particular site by using various carriers, even nano formulations, and this can be also the efficacies can be improved. And the last one is the fifth generation. Here we will see the nano robots, gene therapy, biological system, long term delivery system, that is 6 to 12 months. So, here also we use various nanocarriers as well as the gene therapy, and nowadays we know that by using the CRISPR technology, exosomes, mRNA vaccines. So, this all comes into the fifth generation. So, these are the various generations will scientists have developed based on the limitations of the conventional systems. So, with this we are going to stop today's lecture. In our upcoming lectures, we are going to discuss on the control release drug delivery systems, and also we will see other fundamentals of drug delivery systems. Thank you so much.